everyone. Yeah, beavers. No, I'm not talking about beavers tonight. I'm talking about proteins. Okay, who here has a fizzy drink? Does anyone have a drink that's fizzy? Fantastic. Okay, so what I'd like you to do is, that drink has maybe been in your glass for, what, say, 10, 15 minutes, and it's not flat yet, okay? That's because the reaction between the carbon dioxide in the bubbles and the water in your drink is pretty slow, okay? But those of you who've got a fizzy drink, I want you to take a big mouthful of it, but hold it in your mouth, don't swallow it. I'll start with an experiment, okay? And if this works, you should feel that fizziness of that drink kind of getting more flat whilst it's in your mouth, okay? Has that worked? Yeah? Good. If it has, fantastic. If it's not, that's what it's like to be a biologist. All your experiments fail, okay? So, what's going on in your mouth is that you have this incredible enzyme. This is called carbonic anhydrase, okay? This is in your saliva. And the reason that it's going flat in your mouth is that this is catalyzing this reaction. It's speeding up this reaction between the water in your drink and the carbon dioxide in the bubbles. And it's doing this incredibly fast. It does this reaction 1 million times per second, okay? That's not all of the carbonic anhydrase molecules in your saliva, which is going to be in tens of thousands. This is one single protein molecule doing this reaction 1 million times per second. It's just incomprehensible. So, this, on the other hand, is Rubisco. Okay, this is another enzyme. This also binds to carbon dioxide. Now, this is a bit of an oversimplification of what Rubisco does. It takes carbon dioxide out of the atmosphere, and in plant cells, it converts it into carbohydrates like starch and glucose. Okay, but it is so bad at this. It's so slow. It's an absolute juggernaut. You can see it's actually 16 different protein chains, but it manages to do this reaction only 3 times per second. Okay, it's also got an embarrassing level of specificity because mostly enzymes are really good at being really specific at attaching to just one molecule. We call it the substrate. But Rubisco gets confused between oxygen and carbon dioxide, and that's a real problem because it's a really important enzyme. It's critical in the reactions of photosynthesis, but it's absolutely crap. So here we've got 2 enzymes that are both binding to carbon dioxide, and one of them has the potential to be absolutely transformative because imagine if we could use Rubisco to draw carbon dioxide out of the atmosphere, okay? This could have huge potential for example, solving the climate crisis. So could we take an enzyme like carbonic anhydrase that's so fast and so specific, work out why that is, and engineer that into Rubisco? Or maybe just do away with it altogether and design an enzyme from scratch that could bind CO2. We could solve the climate crisis, we could increase crop productivity with all that extra carbohydrate, we could synthesize biofuels, could be completely transformative. So, I spend a lot of my time thinking about a couple of things. Firstly, proteins. Proteins are amazing. I also spend a lot of my time thinking about some of the biggest problems in the world, and I think proteins have the potential to solve some of those. And we could wait for a few million years for evolution to sort of come up with these solutions, but we don't want to wait. But the exciting news is that we can design novel proteins that have never been seen before in nature, to do tasks that we want them to do. This isn't science fiction. This is happening today. But I just want to clarify, because I've been using the words proteins and enzymes. Enzymes are a subset of proteins. All enzymes are proteins, but not all proteins are enzymes. But enzymes are a really exciting subset because they're these nanomachines that can convert one thing into another, which has got incredible applications for things like biomanufacturing or drug design. And I could just spend the next 15 minutes telling you how cool enzymes are and giving you loads of cool examples. But what I'm talking about today is this whole problem of protein design, not just the subset that are enzymes. Okay. So let's meet our first novel enzyme. And this is one that I'm sure you're all familiar with because the enzymes that are found in washing powder that remove the stains from your clothes are not found in nature. These have been engineered, okay? So these, imagine you work in industry and you say, okay, I want to engineer a protein, an enzyme that is going to remove stains from clothes at, say, a lower temperature, okay? So one way in which you could do this is called directed evolution. Okay, this is a very Darwinian approach to enzyme design. So you've got some DNA, and we'll come to this in a minute, but basically DNA is the code for proteins and therefore enzymes. And you've got, let's say you've got an enzyme that does an okay job at digesting stains at low temperatures, but you want it to be better, okay? So you find the gene that codes for that enzyme, and you deliberately make some lots of copies that are slightly different to that, okay? And then you take those genes that are all slightly different, you synthesize the proteins that they code for, and you screen them. So you get them to do the job that you want. So your user-defined goal here is, I want this protein to digest stains at lower temperatures. You select the best ones, you go through the process again. So this is basically, it's a bit like kind of selective breeding, but on the molecular scale. Okay, so I'll stop talking about laundry because you thought you were just going to have one talk about laundry tonight. I'll leave that to Nick. But basically, directed evolution, this has been used since about the 1980s. It actually won the Nobel Prize for Chemistry in 2018. But the most exciting thing that's happening today in the world of enzyme design involves machine learning. This is this new era where we're able to really play God with these enzymes. So to understand how this works, I'm going to talk a little bit about proteins and their structure. So proteins are polymers. That means they're long chains of repeating units, and these units are called amino acids. Okay? And these amino acids form this chain that then, in the watery environment of the cell, folds up into this complex three-dimensional structure. And what you're seeing here is it's really a bit of an oversimplification, but you can see some of these molecules are orange and some of these are grey. Okay? And this is a bit like kind of protein origami. So you're taking this linear chain, it's folding up. And what's happening here is these orange molecules, if you watch, they're kind of being embedded in the centre of the molecule. And that's because this is all happening in an aqueous environment. These orange amino acids, these subunits, these don't like water. They're called hydrophobic. So you can see that they're kind of being buried in the interior of the molecule, whereas the grey ones are on the outside. Now, that's not all that's going on. There's a lot of sort of fine-scale physics that's tweaking this and getting us to this final complex three-dimensional structure. But that's basically what's happening when this protein is folding. And that final three-dimensional structure that forms is really critically important. It has to be really precise because so many proteins work by binding to other molecules. So here we've got a receptor binding to a DNA double helix. We looked previously at the example of the substrates binding to the enzymes. And this is a bit like if you've got a lock and a key, and your key opens your front door. It's because it's got a specific shape. Okay. Now, I have to emphasize that these blobs here are atoms. We're talking about real atomic scale here. It's the fact that these things are so tiny is why you can get something like carbonic anhydrase catalyzing 1 million reactions per second. We're talking about the atomic scale. So if we want to engineer a new Rubisco molecule, we have to understand this really precise atomic level structure. Okay, so to summarize, we've got our genes on our chromosomes that are coding for our sequence of amino acids. So the different colours here are representing the different amino acids. And that's forming our linear chain of amino acids that's folding up, and that gives us a particular function. Okay? Now, we've known for a long time how to go from here to here. We've been able to sequence genomes for a long time, and we know how this code of A's, T's, C's, and G's translates into this code of amino acids. But the thing that we've wanted to know for a long time is, how does this linear chain fold up into this precise three-dimensional structure here? Now, this is a big problem because there's actually 20 different amino acids, and that chain length can be anything from about 50 amino acids to 35,000 amino acids. So imagine if I gave everyone in the room here a 10-meter long ribbon, and I said to you, okay, roll this up into a kind of ball shape. You'd all do it in a slightly different way, and lots of your structures would unravel, okay? So this is kind of represented in thermodynamics by this thing called a folding funnel. So this is trying to illustrate how big this search space is, how many different possible three-dimensional conformations there could be, but only one of them is the actual stable three-dimensional structure that will form in the aqueous environment of the cell. So, just to recap, we want to know how we're going from this to this, okay? And this is, or rather was, the protein folding problem, because this has been solved pretty spectacularly by AlphaFold, which is a machine learning model that works out how we go from a linear chain. It works out the three-dimensional structure from the linear chain. So scientists and engineers at DeepMind used a large neural network to work this out and published this a couple of years ago. So we can now go from here to here to here. We're really able to predict the structure, and from there we can do experiments to understand the function. But can we go the other way? Can we start with a function, design a three-dimensional structure, and then work out an amino acid chain from that. Okay? This is called the inverse protein folding problem. But can we go further? Could we say, for example, here's the SARS-CoV-2 spike protein, and it's binding to this receptor on the host cell membrane. Could we say, I know the structure of this, because I can use AlphaFold to work out the structure of this, and I want a molecule that's going to bind to it to stop this interaction, to stop it attaching to the host cell membrane. But maybe that molecule doesn't exist in nature. So, can I take that function that I want it to bind to the spike protein. Can I work out a structure that's going to have that function? Can I work out the amino acid chain that's going to give that three-dimensional structure? And then I can work out the DNA code. If I can then put that into some bacteria, get the bacteria to make the molecule, and then use that therapeutically. Now, this bit here, shown with the red arrows, we've been doing this for decades. Okay? This is how we have been able to provide people with diabetes with insulin. We just take the gene for human insulin, we pop it into E. coli, we get it to synthesize loads of insulin protein molecules, and then we can use it therapeutically. But it's this bit here, with the blue arrows, that is the emerging technology that I want to tell you about today. Okay? So I'm going to tell you a bit about the history of this, and then tell you about some really recent research in the field. So, we said that one of the methods that we can use to generate novel proteins is directed evolution. And that's very Darwinian in its approach. What we're going to talk about now is called rational design, and this is the kind of creationist end of the spectrum. This isn't that new. This has been done since this was the first fully novel protein to be synthesized that was designed with a computer. It's called Full Sequence Design 1, and it had a structure that hadn't been seen before in nature. That was in 1997. And then in 2003, Top 7 was synthesized, and this had structural features that had never been seen before in nature. But the field has absolutely exploded in the last month. I signed up for this talk in August. I did not anticipate that there would be such an explosion of research literally in the last month. Because as we saw, AI has absolutely revolutionized protein folding, and it's now absolutely revolutionizing protein design. Okay? Now, I want to make something really clear. I am not a computer scientist, okay? But I'm going to do my best to tell you a little bit about the science behind this. So there's been a lot of excitement in machine learning about something called diffusion models. So the way that this works is that you generate training data for your machine learning model by taking clean images and gradually adding noise to them until they become pixelated, okay? So you're going in this direction. And then what you get the machine learning model to do is to take that noisy image and go the other way and get it to extract this from the pixelated data, okay? Then that's called denoising, going in this direction, okay? Then if you want to get this model to generate something new, you give it some pixelated input, and you give it a prompt. So let's say I've asked this model to generate me an image of a face. And because it's learned from the training data how to do that, and it uses that prompt, it gradually creates the image from that, okay? So, you've probably seen this online because you might have heard of DALL-E 2. I'm a massive fan of this genre of kind of like modern problems in a historical art style. This has been insanely successful in the 2D image space. Okay, but you might be thinking, well, what's this got to do with proteins? Proteins are three-dimensional. Well, can we go from 2 dimensions to 3 dimensions, and instead of using diffusion models to generate 2D images, can we ask them to think about a 3-dimensional space and the coordinates of atoms in space? Well, the answer is yes, and this guy is leading the way. This is David Baker, and he is the director of the Institute for Protein Design, the University of Washington. And his team were actually behind Top 7, that was that engineered protein that we saw that was developed in 2003. And it's the paper that he published last month that made waves. Okay, so instead of giving DALL-E 2 a caption and getting it to make a 2D image, he said they used him and his team used a diffusion model. It was a different diffusion model, but they said, okay, design me a protein that can bind to this molecule, or design me a protein that is an enzyme that can catalyze this reaction, or design me a cage that has 8-fold symmetry. Okay, these are called design constraints. So what we're talking about here is being able to do this blue bit of this sequence. Okay, so this is how it did. This model is called RF diffusion, for Rosetta fold diffusion. And this was where they asked the model to, going back to our spike protein, they said to the model, okay, design me a protein that can bind to these 3 symmetrical ACE2 binding sites. So this is the ACE2 binding site that's shown here. And here you can see the diffusion model generating this structure. Now, if we go back to thinking about prior to using models like this, using something like directed evolution, you would have had to screen thousands of different molecules until you found one that did the job that you wanted. But when they were doing this, they were having to look at just potentially 1 molecule per design challenge. So they're basically getting proteins to do jobs on demand. It's incredible. So you might be thinking, well, so what? This is all just being done in a computer. What's the kind of real-world application of this? Well, they went one step further. So they took those amino acid sequences that had been generated in the RF diffusion model, and they synthesized them in a wet lab. So they've verified that these structures do fold as intended. So this is an electron microscope image of this protein that was designed. And these nanocages are potentially going to be really, really important in vaccine design. But we haven't verified that these things are safe. We haven't verified that these don't bind to all sorts of other things in the messy environment of the cell. And it's really worth emphasizing that this research was published online on the 1st of December and then updated on the 14th. It was, hasn't even gone through peer review yet. Okay, so I'm not saying that this is going to solve the big problems that I mentioned. I'm not saying that because of this paper being published, we can redesign Rubisco. And it's not even that machine learning is the only option. So there was an interesting paper that was published last year that was looking at enzymes to digest plastics, novel enzymes, and they didn't even use machine learning. But I think this is really exciting, and I hope you do too. Thank you.
Thank you very much. Jenny is now testing the bubbles theory.
Still fizzy.
True biologist doing the experiment. Do we have any questions? Any questions from the crowd?
No? Okay, bye. Hello.
What's one way in which this could go horribly wrong?
Oh my gosh, yeah. What's one way in which this could go horribly wrong? Okay, so yeah, this could definitely be used by some bad actors. I think something we've seen in biology is that technology that has been kind of like Nobel Prize winning a decade ago can now be done by people in their basement. Okay, so you look at something like CRISPR-Cas9. So this came on the scene about 10 years ago. You can now buy a kit for this online and sort of do it in your basement. I don't know if we're going to see the same thing here, but there is this acceleration in biotech where policy is just kind of not keeping up with how fast this is moving in research. Yeah, there's definite potential for this to go quite wrong. So actually, I was reading about how some of David Baker's early work was funded by DARPA. That's not to say that there's anything wrong with him, but clearly, like, defense organizations are interested in this. And yeah, some proteins can be very, very nasty. So who remembers mad cow disease and CJD and BSE? So that disease has 100% fatality. Luckily, it's very rare. But that disease is weird because it's infectious, but it's not caused by a pathogen. It's not caused by like a bacterium or a virus. It's caused by a protein. And the name prion actually stands for protein infectious particle. So, yeah, could we use this to engineer some prions that could potentially... they work in a really interesting way, by the way, just going back to the kind of protein folding. Prions cause proteins to have, where we saw those kind of like spiral helices, they cause other proteins in your cells to refold and for those helices to turn into these kind of flat structures, and that causes kind of chain reaction. So there's some really weird biochemistry. Nobody really has worked out how it all works or how to treat it. But yeah, there's definitely some potential for designing that, or maybe designing better, I say better, more infectious viruses. But yeah, I think the existential risk of engineered pandemics is a talk for another time. Hi.
Links to that. In the research paper that was published,
Yeah.
have they published how they sort of the methodology of how they've of the AI that they used, and could somebody else take that methodology of the AI that they use? I'm not sure, you should probably tell them.
Well, I'm not a computer scientist, so.
Could they take that, bad actors take that and go, I'll do this, or I'll do this?
Yeah.
Similar in the way that somebody might sort of synthesize something like the X gas using AI or something.
Sure. Okay. So the question was, could we take the model that could somebody access the RF diffusion model that they used and use it? I don't believe it's like open access. I don't believe you can use that one yet. But going back to AlphaFold, all of their protein structures were published. Well, almost all of them were published online in the last couple of years, and the ethics team at DeepMind did do a lot of work sort of thinking about this really carefully and saying, okay, could this speed up the development of, say, engineered pathogens? On balance, they came out and said, actually, the benefits of doing this outweigh the risks, but that's always going to be a balance. But then if we have these models, then if somebody does engineer a new virus that causes the next lockdown, you can do some more knitting maybe, at least we can then fight fire with fire and maybe create some antibodies or drugs that could bind to that. So that's potential. Yeah, fingers crossed. Yeah, exactly.
I'll take your measurements during the break.
Awesome. Yeah. Thank
we
you.
actually have a question from someone watching us at home. Is folding home with AlphaFold obsolete? Is the question.
Is what? Sorry.
Is folding at home?
Folding at home. Okay.
Obsolete.
Oh, this goes back to my first Nerd Night talk, which is about citizen science, which was all about how you. So folding at home is like a distributed computer model where. Does anyone remember SETI at home? Yeah, so where you ask people to run something on their computers that involves a lot of compute. Yeah, I think it basically does. But there's a really cool game called Fold It that is where you can actually try to fold proteins. And there has been some, actually David Baker's lab was behind Fold It and Folding at Home. So yeah, it's worth playing with.
I saw another question over there, yeah.
Hello.
So towards the end, you can use it to other non-AI ways of doing
Yes.
I'm guessing you might have wanted to talk about that before the paper came on.
Yeah, so directed evolution is one of them, but that's a pretty blunt instrument because actually directed evolution doesn't require you to have any knowledge of protein structure. You can just say, here's a protein that has a function, I want the function. You just need a way to screen for the function. You don't actually need to kind of like know anything about the structure because you're just testing whatever user-defined goal that you have. You can also, if you have knowledge of the protein structure, you can try to tweak it by mutating the DNA so that certain amino acids change. So for example, with an enzyme, the bit of the enzyme that's really critical is called the active site. So that's a bit that the substrates bind to. So you could try through a sort of trial and error of like modifying those amino acids that are in that active site and seeing if that has an effect. So yeah, there are other methods that don't have the same kind of success rate. Yep.
So do you have like a backlog of things you wish you could make? Do you know what I mean? Is there
Me personally.
What are you personally? Yeah.
I hadn't thought about that.
Make this or something to do that, something to do that.
I mean, I'd quite like to solve the climate crisis. That would. So I'm really interested in alternative proteins. So these are proteins that could be replacements for animal products. So I think this is a really exciting field of where we can so instead of eating something like an Impossible Burger that's made from plants, maybe we could engineer the kind of proteins that are found in meat, but engineer them outside of animals, so we could do away with factory farming. Perhaps this has got an application there. So I think that's top of my list. Hello.
How long did the process take? You have to do error for one.
Yeah.
Because I remember when that technology started, it took time.
Yeah.
From the sequence, even if it's a little wrong with the sequence,
Yeah.
you can change everything.
cool. So the question was, how long does that process take? The bit that I showed with the blue arrows. So with the RF diffusion model, like minutes to hours, like it's insanely quick. You're literally typing in, and I don't know if any of you have played with DALL-E, or DALL-E 2, sorry, and just type in a prompt, and it basically generates it there and then. The speed is phenomenal. Yeah. Oh, I'll go over here. Hi.
So going back to the climate crisis, you
Yeah.
used the idea of kind of making photosynthesis much more efficient.
Yes.
Is anyone actively working?
Yeah. Yeah. Is anyone working on making photosynthesis more efficient? People have been interested in this for a long time because basically Rubisco is the only sort of biological method we have of drawing carbon out of the atmosphere and sequestering it. And there's been a lot of interest, for example, in there's a type of photosynthesis. Because Rubisco is so crap, plants have evolved this real sort of workaround to get around the fact that it's so rubbish. So there's a couple of types of photosynthesis. One's called C4 and one's called CAM. And these basically, so I said that it gets confused between oxygen and carbon dioxide. So one of the ways that plants can get around that is that they can try and hide the Rubisco away in a location where it's saturated with carbon dioxide and try and keep the oxygen out. And that makes it work better. There's also been some interest in kind of like engineering some of the enzymes that activate it as well, and increasing the concentration of a molecule it binds to called RUBP, which is one of its substrates. But there's been a lot of interest in trying to engineer the C4 sort of method of photosynthesis into things like rice. There's someone called Jane Langdale at the University of Oxford who's trying to engineer C4 photosynthesis into normal plants, which are called C3 plants. That's actually more for kind of like crop productivity, but there's no reason why that can also be for carbon sequestration. But actually, the increasing carbon dioxide concentrations in the atmosphere are speeding up photosynthesis. So it's called carbon fertilization. But yeah, clearly not at the speed that we need it to get us down to safe levels. Hi.
Hi. Do you have a library of the proteins and the function, the proteins that you need to match the function that you want?
Okay.
The three green protein structures that we saw in the picture. Were they random, or do you have can you know how to match those with the function?
Yes, the question was, is there a library of those proteins? Yeah. It's called the Protein Data Bank, and it's really cool. So all of these structures that have either been found out experimentally using things like X-ray crystallography, or with computers using things like AlphaFold. You can go online and you can find all of these structures online. So that's probably where this came from here. Yeah.
Last question. There was one more hand.
Yes.
before.
Hi.
Hi. Really good talk.
Oh, thank you.
I think I got the impression that all of the searching for new proteins and stuff is pretty much give it to an AI model and see what comes out. Is there any theory, or is there going to be any theory in the future that will help us understand and maybe speed up the AI process?
I don't know about speeding up the AI process. I mean, people have been interested in finding enzymes just in nature for a long time. So there's been a lot of work to just kind of take gallons of seawater or loads of soil and just kind of search through it and find enzymes that way. But yeah, I'm not sure about speeding up the AI side of things. It's pretty fast. Is it not fast enough for you? I mean, things are moving pretty fast, and not just in the research space, but actually in silico as well. So exciting times. Can we squeeze one more in?
Yes, absolutely.
Hi.
Have you been given a structure? How do they test the structure actually delivers the function?
Yeah, great question. Having been given the structure, how do you test that it actually does what you want it to do? So that's kind of the next steps, because just saying that, okay, we've got a molecule that can bind to these, and we can maybe show that by getting cells to synthesize this in a laboratory and testing it in a cell culture. That's very different to then testing it, say, in a human body, okay? Because we're a very messy mixture of biochemistry. So there's also some interest in kind of trying to use AI to speed up that kind of clinical trial process, but you're still going to have to check the safety, because there's also a lot of interest in trying to work out how these proteins could bind to other proteins, because you don't want to synthesize this and then find that it binds to, I don't know, your DNA polymerase, which is like a critically important enzyme in your cells. So again, there's some AI research into trying to look at how proteins bind to other proteins and try to predict this. But at the end of the day, I think you have to actually just try it out in an organism.
Which is a great place to leave this
There we go.
at. Thank you.